Okay, we'll get started. Um, Thank you for joining this session in the Networking Lounge. I'm Chris Stamen, Senior Program Officer on the Maternal Neonatal Child Health Discovery and Tools team at the Bill and Melinda Gates Foundation. I'm joined by Srivali Krishnan, Senior Program Officer in the Agricultural Development team in Southeast Asia. And I'm super excited to delve deeper into the fermented foods and discuss the grand challenge on evaluating fermented foods in the context of maternal nutrition. We'll have a series of 4 speakers with rapid talks. Please keep your microphones muted during the presentation and feel free to add your questions to the chat. If you like a question that you see, like it, and we will use this to prioritize. At the end of the presentation, we'll have a Q&A moderated by Shelby Montgomery, Senior Program Officer, and Brendan Thompson, Program Officer in the Discovery and Translational Sciences team. Okay, without further ado, let's get started. Turning things over to you, Srivali.
Thanks so much, Brendan. Hello, everyone. As Chris mentioned, I'm a Senior Program Officer with the Agriculture Development Team at the Gates Foundation. When we talk about fermented foods, I just wanted to give a very brief introduction and have my colleague Chris actually take into a little bit deep dive around some of the discovery as well as on the translational science. Can I have the next slide, please? If we look at the multi-sectoral effort to improve nutrition, we understand that food is an important component when we are addressing the nutritional challenges moving forward. The maternal and child undernutrition can have both short-term as well as on long-term consequences. But it is important that when we are looking at the basic causes and underlying causes, we actually look at how we are using multi-sectoral approaches to really address some of these challenges moving forward. Good nutrition also depends on a lot of practices, but also in terms of a behavioral change. And also it is important that the diet quality is very critical and fundamental when we're talking about improved nutritional status, particularly of very important target groups. Several micronutrients and macronutrients are necessary to maintain the good nutritional status. And to do so, we are also seeing that there is importance of fermented food in this sector. The next one, please. When we talk about the history of consumption of fermented foods, and I just wanted to give a very brief glimpse of what happens in the Asian context. Here we see that from the north to the south to east to west, traditional Indian or traditional Asian foods have been fermented since 1000 BC. So traditionally, fermented foods have been included in the diet, and these have been consumed by traditional communities for a very, very long time. Some of the foods that we are now seeing in a lot of Asian markets or also in Asian restaurants around the world are also the product of fermentation, but also they are producing enough micronutrients that is important for the improved the gut microflora moving forward. Can I have the next one? When we talk about the traditional foods, and going back in the history, why were these fermented foods introduced? There are several reasons to plot with, but few of them have been listed here, which is basically the relatively simplicity of the processes and how a household could preserve the nutritional status of these foods over a longer period of time, but also making sure that these are available in the off-season period and around the year as well. Another one was also to basically preserve the nutrients in these food products for a longer period of time, but also improve the flavor, the digestibility, and overall nutritional benefits. While we are all aware of multiple types of fermented foods, such as pickles or different types of yogurts that we are generally consuming nowadays, it is important to understand that these foods have traditionally played an important role in not only improving the nutritional uptake of the individuals, but also in helping in the kind of palate that is being built over particular communities over a longer period of time. And these palates have been very, very important from the cultural standpoint, from the traditional standpoint of what a community has traditionally consumed and what the community is used to consuming over a longer period. There are several categories of fermented foods, which include cereal-based, cereals which have an addition of milk, cereals which are in the form of sweets or savories, several fermented foods which have vegetables, pulses, and different types of condiments that are added to make sure the fermented foods are preserved over a longer period of time. It is important that when we are also looking at fermented foods, the delivery and how these foods are actually delivered to the target population is also critical. Several times the composition of the fermented foods, for example, when we are talking about pickles, there is a higher salt or an oil concentration, but also it is a point where the fats and other aspects of the foods are not processed. They are available in the raw form, which at point is sometimes very beneficial for absorption of those nutrients. Can I have the next one, please? When we talk about where are the opportunities for scaling up of availability of these traditional and fermented foods among the population and the target population with whom we are really concerned with where to address the various challenges of undernutrition, it is important that when we are talking about cutting edge technologies from crop to farm diversifications, we always take a critical but at the same time a longer term view. It is important that we are looking at transformative approaches that are both innovative but also that have a model of being scaled up both by the public as well as from the private sector perspective. This will not only help in the research and discovery, but also in improving access to these products and services over a long-term period. Now, when we are talking about taking these partnerships to scale, I would like to also look at opportunities, particularly when we are talking about how these roll out in the country systems. How can we introduce these over the large scale programs that the public sector is handling in several countries, whether it is the take home rations or it is the public distribution system or in the ICDS or the schemes around school feeding programs, particularly when we are talking about the target population. It is important to note that the traditional fermented foods vary from region to region, and they're also there's a point of how you're introducing and the point of delivery of these and making sure that they are achieving at scale. With these strategies, it is important that we are working both with the academic, the research, the civil society, also the public as well as private sector in partnership to make sure some of these are available going forward at scale, but also some things that can be introduced in large scale programs across those countries. Can I have the next one? When we are talking about scaling up of efforts, it is important to note that how can we introduce these and make sure that we promote the consumptions over a critical life cycle period, and also look at behaviors that can promote and inhibit consumption of fermented foods. As I had mentioned, there are traditional knowledge. There are obviously certain palates and behaviors that are exhibited in different communities, and how are we making sure some of these activities can actually look at how they can be promoted and rather not inhibit the consumption of these fermented foods. And also it is important from the affordability, from the point of which how we can convert the household production to consumption of these fermented foods, and how can they be done very simply, and also follow the processes that can be adopted over a market accessibility as well as local availability standpoint. Over to you, Chris.
Thanks, Shivali. That was wonderful. Really, thank you. Such a rich tradition of fermented foods in Southeast Asia and really valuable perspectives. So as a gastroenterologist from the US, I can't begin to understand the topic with this richness. And I approach the topic with humility, but wonder if there is a value in a few additional framing slides. So Brendan, please just go ahead and advance the animation here. So this first slide is the growth and resilience framework. That's one of the cornerstones of the maternal neonatal child health discovery and tools strategy. There are 2 points I'd like to draw your attention to on this slide. So first, data supports that intervening earlier in the life cycle may be most impactful for both maternal and pediatric outcomes. One more advance, Brendan, or one more. Perfect. And second, we hypothesize that there is a tenacious underlying biological vulnerability that's a common thread and responsible in part for poor birth outcomes, infections, poor pediatric growth, and neurocognitive development. So next slide. So what is that underlying vulnerability? As a gastroenterologist, I see the world through GI colored lenses, and quite simply put, one might say that we're simply tubes. So next, Brendan, you can advance to the end here as well. In health, back one, please. These tubes are the ingenious product of eons of evolution from worms to Homo sapiens that serve as biofactories for transforming ingested foods by host and microbial factors into more nutritious products that can be absorbed by the body. So for example, a healthy microbiome produces B vitamins and captures as much as 10% of the calories and undigestible fibers that would otherwise be lost out the other end. And in disease, the food is lost to maldigestion, malabsorption, inflammatory consumption, inefficient metabolism. And the key insight here is that it's not just about the right foods, but also making sure the gut and the microbiome organ are working properly to transform the foods. So next slide, please. So there are some great insights from antiquity and lessons that we would do well to embrace in modern medicine. And Hippocrates wrote, "Let food be thy medicine." Next. And the gut perspective in modern technology teaches us that we would do well to add microbes to that equation. Interventions that address not just the issue of undernutrition, but the disease process occurring in the gut will lead to more resilient and durable and ultimately impactful interventions. Next slide. So this is the space where many of our discovery and tools interventions fit. Next slide. We have microbiome-directed therapeutic foods and gut microbial therapeutics in the pipeline. And fermented foods offer a complementary approach that embraces evolution, culture, and proximal partnership. It brings food and microbe together outside the body. It transforms less nutritious foods into those which contain a rich array of microbes, vitamins, micronutrients, and other bioactives. So as we'll see in the next talk, this biotransformation can lead to improvement in microbiome diversity and systemic inflammation. It's a pleasure to introduce to you Erica Sonnenberg, a senior research scientist in the Sonnenberg Lab at the Stanford University School of Medicine. She's co-author of The Good Gut and investigator on the fermented food trial that we'll see next. Erica?
Thanks so much, Chris, and thanks for the opportunity to present a clinical trial that we recently performed in our lab, in which we looked at the impact of fermented food consumption in a human cohort on inflammation and diversity of the microbiome. Next slide, please. So what the past 20 years of research has demonstrated is the profound connection that our microbes have to our health. The composition of the microbiota or microbiome largely dictates the way that we respond to an infection, even if that infection is in our lungs, for example. It also dictates in many ways our metabolism. So the calories that you ate today, are those going to be burned or stored as fat? That is dictated by the microbes in our gut. And then more recently, there have been indications that there are also profound connections between our gut microbiome and our central nervous system. So perhaps moods and behaviors can in fact be dictated in many ways by the microbes within our gut. So this profound connection that exists between our gut microbiome and our health provides an opportunity from a therapeutic standpoint where we could use our knowledge of this connection to manipulate our microbes in a way to improve our health. And one of the things that we have learned is that diet is in fact a very powerful lever in controlling our gut microbiome. And so the question that we had was whether we can manipulate our gut microbiota to improve health through the use of diet. Next slide, please. So the study that I'm going to talk about today, we recruited 18 healthy individuals, and these individuals were instructed to slowly ramp up over a period of 4 weeks their consumption of fermented foods. Then for 6 weeks after that, they were instructed to eat as much fermented foods as they possibly could. And then for 4 weeks after that, they were instructed to make whatever choice they were comfortable with. They could continue to eat high amounts of fermented food or not, as they decided. And during this intervention, we took several stool and blood samples longitudinally, and we used these samples to perform omics analysis to give us an understanding of both changes in the microbiome that were occurring during this dietary intervention, as well as changes in immune profile. And the omics data that we generated was critical in understanding how fermented foods were impacting both the microbiome and the immune profile, since we really didn't have a good understanding going into this study what sorts of changes we should expect. And so accumulating these vast quantities of data really helped us to understand how fermented foods were influencing participants' microbiome and immune system, and will allow us in the future to generate more targeted hypotheses for what we would expect fermented foods to do. Next slide, please. So participants were allowed to eat whatever fermented foods they were comfortable with, and they chose a variety of fermented foods, anything from kombucha, yogurt, kimchi, these kinds of foods. Next slide, please. So what we found was that participants were actually very effective at increasing the fermented food consumption during this intervention. So at baseline here in the bottom left, participants consumed on average just about a half serving of fermented foods per day, which is pretty typical. They increased that consumption over the ramp period, and by the peak of the maintenance phase, the participants were consuming on average about 7 servings of fermented foods per day, which was pretty amazing. Next slide, please. So when we looked at what was happening in these participants' microbiome, what we found was that participants had an increase in the diversity of the species that we were able to detect within their gut. So this graph here on the right shows the number of total species that we detected in the gut on the y-axis, and then time on the x-axis. And as you can see from this line drawn through the average diversity that we observed in these participants, there was a steady increase in the total number of species that we detected within the gut of these participants. And so if we just take this at face value, one thing that would appear obvious is, well, these people were consuming bacteria. Of course, you would find more bacteria species in their gut because they were just eating more microbes. And so we wanted to test whether that was the case directly. Next slide, please. So we went out and purchased all the fermented foods that these participants had consumed throughout the study. We struck them out on plates to see if they were viable. We picked many, many colonies from the fermented food plates and identified the microbes that we were able to detect within the fermented foods. And then we asked the question, are these microbes that participants were consuming responsible for the increase in diversity that we saw within their microbiome? Next slide, please. And in this pie chart here, what we show is that only a small sliver, so the little sliver in peach here, is the contribution from the microbes that participants actually consumed. So only about 5% of the new species that we detected within their guts actually came from fermented foods. And 95% of the new species that we detected actually came from some other source. And so it appears that fermented food has an indirect effect on microbiota diversity. There's something about consuming fermented foods that allows people to gain new diversity within their microbiome. And we think this is important because increased microbiome diversity has been shown to be beneficial broadly for health of humans. Next slide, please. We wanted to see how fermented food consumption affected these participants' immune status. We measured many inflammatory cytokines and chemokines, and this figure here on the left shows several of the cytokines that we measured, and they all went down in amount over the time of the study, indicating a broad decrease in inflammatory cytokines for participants. Next slide, please. We also looked at different cell types and the amount of activated signaling proteins found within those cell types. So this graph shows difference from baseline to the maintenance phase when they're consuming a lot of fermented food. And we saw a general decrease in inflammatory signals in participants in this study. Next slide, please. So what the study demonstrated was that gut microbiome targeted diets can be an effective, scalable approach for improved health across populations. In our cohort of healthy adults, we found that fermented foods increased gut microbiota diversity and decreased markers of inflammation. Next slide, please. And so we're questioning what the yet unrecognized health benefits of fermented food might be. We think that there may be broad benefits to many different population types, but we will need more well-designed human trials that employ these omics technologies so we can really get a broad understanding of how fermented foods influence human biology. Thanks so much.
Erica, that was wonderful. Thank you so much for providing details on this quite landmark trial. Small, but I think as a harbinger of some really interesting work to come. You show that there is impact on microbiome diversity and on a decrease in systemic inflammation. And I'd like to introduce Najiha Iqbal, who is associate professor with a joint appointment in the Department of Pediatrics and Child Health and Biological and Biomedical Sciences at Aga Khan University. And her research is also focused on biomarkers, but as they associate with growth faltering and malnutrition in children. And super excited to have her present on these very early data, look at this data in the Vital Pakistan Trust lactation study. Najiha, over to you.
Thank you, Chris. Thank you so much for this opportunity to present on behalf of Vital Pakistan. So I'm presenting a salient result of the trial, which was a nutritional-based supplement trial for lactating women with or without azithromycin for infants, and to associate the outcome in the growth of children at 6 months. Next, please. So just give you a brief methodology of the trial. It's an open label, community-based, randomized controlled trial. And the criteria for enrolling lactating was mother within the first week of birth, having walked less than 23. Trial design and procedure was first is the screening and the eligibility after informed consent. Subjects were randomized into 3 arms. The arm A received nutritional counseling and exclusive breastfeeding and newborn and infant care. The arm B receives counseling plus nutrition supplement given to mother at the time of enrollment till up to 180 days. And the arm C was nutrition supplement given to mother, as well as azithromycin single dose was given to infant plus counseling. And the intervention that they used was locally produced, ready-to-use supplement, which is basically based on chickpea and peanuts and skim milk. And they 2 sachets were given to these women until 180 days after the enrollment. And for the azithromycin trial, 1 single dose of azithromycin was given to a child at day 42. Next, please. After enrollment, the trial procedure was defined in this slide. So for is a continuous follow and on the breast call and the compliance, and which was on daily alternate 72 hours and on weekly basis, as well as there is a serial anthropometric measurement of both mother and the child at baseline, 27 days, at day 56 up to 180 days. In terms of sample collection, they have two. They have two schedule of sample collection. One is day 41 and day 56, and infant blood was drawn for assessment of the hemoglobin, transferrin, ferritin, proteomics, and other inflammatory biomarkers like AGP and CRP. And the breast milk was collected from mother. Maternal blood and maternal infant stool was also collected at two time points, which is 41 and 56 days. And different the biomarker assessment were based on assessment of macro and micronutrient from breast milk, HMOs, and microbiome from breast milk, and maternal blood. In maternal blood, the same biomarkers were assessed as assessed in the child, like ferritin, transferrin, other inflammatory biomarkers, and from the stool, they basically look for the enteropathogen, B. infantis, and other two inflammatory biomarkers which are important, calprotectin and lipofilin, which is emerging as a new biomarkers. They were also assessed from the infant and mother stool. Next, please. So the rationale and the aim of the biomarker analysis from the fecal sample was to observe the difference between the maternal and infant biomarkers, to understand the difference in baseline state between maternal and infant biomarkers. Secondly, to determine which biomarker associated with a good clinical outcome, which is based on the anthropometric measurement and the status and pediatric growth and neurodevelopment outcomes. And also to determine the relationship between biomarkers in different study arms, and basically to understand the association between biomarker and the clinical effect observed in different intervention arms. Next, please. Yeah, so in this slide, basically the two biomarkers were, two fecal biomarkers were assessed in infant and mother. So first is calprotectin here. So you can see the blue bars are infants, and the red blue and the red are basically showing the different time points. So the blue is the age at 40 days sample collection, and the red is the 56 days. And the first first is the infant analysis, and then the mother analysis. So you can see the calprotectin was low. It was not very significant, but was very low compared to infant sample. So if you can compare the baseline, which is the 40-day, you can see the calprotectin level is very low in mother compared to infants, and same as for 56-day. The second biomarker is lipofilin, which is a marker of intestinal injury. It has shown some association in acute kidney injury. So it's one of the emerging inflammatory biomarkers, which is also shown in different cohorts of different malnourished cohorts. So you can see that in infants there is a low level of lipofilin, but in infants, but compared to mother there is a high level of lipofilin at both time points. Since this analysis is not comparing arm-wise analysis is not yet done, so we cannot associate we cannot conclusively say something about the role of different treatment that was done in these arms. But what we can see here is that there is a trend of increased lipofilin, which is a marker of intestinal injury, and it's a marker it's indirectly show inflammation is higher in mother compared to infants. But for the calprotectin, which is a marker of a neutrophil infiltration in fecal sample, it is higher in infants compared to mothers. But it is not statistically significant, and I think the trial-wise analysis will clearly give us the picture of these biomarkers. Next slide, please. Then we looked at the pathogen burden in the fecal sample. So these pathogen burden association, this pathogen detection was done by TACMAN arrays, a real-time PCR, which basically is which is a multiplex PCR and which detects bacterial, viral, and protozoal targets. So if you see here, the blue bars basically show the first time point, which is a 41-day, and the yellow on, is the infant time point, which is a first. So if you look at the first figure, which is at day 41, and compare it with day 56, so we can't much appreciate the change in the pathogen burden, because those pathogens which were higher at 41 days are still higher at 56 days in both mother and infant sample. And there is no significant difference in both time points. So, but again, the trial-wise analysis will definitely gives us an idea about the role of azithromycin, on the pathogen burden. But still, some of these targets are higher in kids, but it's not really significant here. Next slide, please. Then we next looked at the Bifidobacterium infantis and the longum, the presence of Bifidobacterium longum and infantis in mother and child samples. And this analysis was also done in the fecal sample. And you can see here that the B longum is higher. B longum is higher in both first time point and the second time point, while B infantis is higher in the kids. So this, which makes sense, because B infantis is supposed to be higher in kids. So which makes sense, but again, this requires some deep analysis, which basically compares the arm-wise analysis can basically clearly tell us about the role of different supplementation on the B infantis detection. Yes, please. Yes. This is the slide from the Sonnenberg lab, and what I just wanted to show you is that you can see that the infant are shown as the pink dots here, in the principal component analysis shows the pink spot for the infant, and the mother is blue. And what you can clearly appreciate is that the microbiome clearly separates between a mother and infants, and there is a very minimum overlap in the microbiome. So that clearly tells that the mother has a different microbiome compared to infants. This work is still going on, and we will basically look for the outcome of. We will compare the microbiome analysis with the outcome of the growth of the child. So this is a continuous analysis. Next slide, please. So in conclusion, and what are the next step, and what are the conclusion of this presentation? There are key differences in the enteropathogen load inflammatory markers and microbiome profile in mother and infants. Future analysis will focus on determining which maternal biomarker predict good clinical outcome in infants in terms of growth and neurodevelopment associated with better maternal health. Future analysis will also focus on arm-wise analysis, as I mentioned, that basically it will be more conclusive to discuss about the role of these inflammatory biomarkers. And these analyses will consist of a hypothesis-driven approach using multiple linear regression as well, hypothesis agnostic approach using supervised machine learning. So some deep analysis will be carried out to basically understand the role of these biomarkers and the anthropopathogen burden with overall clinical outcome, both in mother and infant. Thank you. Next slide.
So thank you so much, Najiha. That was really fabulous. And you and the whole VITAL team are really, truly leading the path in exploring the black box of the maternal gut and how that links to both maternal outcomes and pediatric outcomes. Really important work, and thank you for sharing it. Look forward to seeing the data and its analysis as it unfolds. Next, I'd like to, thank you. Next, I'd like to turn to Ravi Sheth. Ravi was a Gates Fellow and is a Hertz Fellow and was at the foundation a few years ago. He's now co-founder of Kingdom Super Cultures, having finished his graduate work at Columbia. He's an extraordinarily bright thinker, as demonstrated during his summer at the foundation, and really honored to have him present in more detail the actual Grand Challenge call and what it entails. Ravi?
Thanks, Chris, for the kind introduction. So yeah, I'm really excited to talk through a little bit of the background and then really dive into the details of this Grand Challenges call around preserving culture. Next slide, please. So to provide some context, and we've already been talking a lot about this in multiple talks today, fermentation is really this ancient practice that's been deeply kind of rooted and intertwined with human biology, evolution, and diet. And if you think about it, nearly every single iconic food you can think of is fermented. And across many different cultures, things like yogurt, beer, cheese, wine, fermented grains, almost all of these foods are simply combinations of kind of raw plant-based substrates plus microbes. And what's really cool about these fermentation processes is that it's really an example of 1 + 1 = 3. You're not simply taking, some sort of microbe and combining it with the food, but these microbes have the ability to go in and unlock and create entirely new types of foods, unlocking nutrition, unlocking taste and flavor, really creating something that's really not just the sum of its parts. And as Justin was talking about earlier today in the plenary talk, this is something that's really deeply intertwined with human biology. Over the course of human evolution, this is a kind of core process that humans have kind of adapted to and has been a core part of both our biology, but also our culture and how we think about food. Next slide, please. And so microbes themselves have this really wondrous ability to improve the qualities of food across many distinct axes. This starts with really basic things like the stability of foods, things like reducing the pH, improving the inherent stability of a food, but also producing targeted bacteriocins, targeted antibiotics that might exclude pathogens, and removing simple sugars that might inherently increase the stability of a food matrix itself. But these microbes, beyond just creating kind of stable, long preservative foods, can also do things like improve the nutrient quality of these foods themselves. And so many microbes found in fermented foods have been shown to produce key vitamins, things like B and K vitamins, and also remove bad nutrients as well, removing and degrading things like mycotoxins, phytates, things that actually kind of directly affect the bioavailability of really key minerals in the human diet. And finally, beyond kind of these technical properties of foods, fermentation itself has this ability to really transform and improve the taste, flavor, and texture, all of these organoleptic properties of foods that can take really basic staple crops and make them taste like something amazing, really something that's not just the base crop itself. Next slide, please. And so particularly in these challenging times with COVID-19, there's this promise of using biology as a really scalable and distributed decentralized manufacturing technology that's really quite exciting. If you think back over the course of the last 100 years or so, much of food processing has been really kind of created and scaled through really centralized chemical-driven food processing supply chains. Specific molecules, things like antimicrobials, antibiotics, antifungals, surfactants, emulsifiers, really need to be produced at really large industrial scales and then distributed out in really complex supply chains to all over the world, nationally, domestically, to power the production of many different types of foods. But what's really exciting about using biology or using fermentation as a manufacturing technology and processing technology for food is that it's uniquely scalable. At the end of the day, microbes themselves can self-replicate, and this represents this really incredible kind of magical technology that can be kind of shipped all across the world and really power the decentralized production of food in a way that's currently not possible. Next slide, please. So despite the promise of all of these kind of fermentation approaches, one key challenge is that almost all traditional fermentation processes themselves remain highly uncharacterized. And so if we look to many of the different fermentation kind of traditions and cultures that have been established across the world, there's a very few number of these that have truly been studied, scaled to large-scale industrial production, and the individual microbial actors and the nutritional benefits and the health benefits truly characterized in a really quantitative and deep way. And so there's a rich history around plant-based fermentations, things like adding salt and lactic acid bacteria to specific vegetable substrates to ferment them. But even beyond these well-known kind of sauerkrauts, the kimchis of the world, there's a huge amount of kind of fermentation processes that really have not been studied to the same depth. There's a whole kind of class of fermentations leveraging fungal organisms, things like aspergillus used to make koji, rice fermentation, many different types of grain-based fermentations that have really been studied to a very small degree. And one thing that's interesting to highlight is that in many cultures, many fermentation processes themselves aren't specifically considered to be fermentation. Things like letting grains soak overnight in water, this is something where there's actually living organisms growing and fermenting these substrates, improving the bioavailability, synthesizing of different nutrients, synthesizing specific nutrients. But in many cases, some of these fermentation processes are not considered fermentation per se. It's simply just a part of preparing food at the end of the day. Next slide, please. And so with this background and context, really the key idea behind this Grand Challenges call is enabling and the study using modern technological tools, using microbiology, using sequencing, using in-human clinical trials to really rigorously evaluate and study and understand the kind of potential human health benefits of many of these traditional fermented foods. And the real focus is enabling local investigators in specific geographies to take traditional foods and traditional fermentation processes of interest and study these using these next generation microbiological and microbiome techniques. Next slide, please. And so very specifically, this Grand Challenges call seeks to fund specific pilot studies that investigate the biological effect of traditionally and locally fermented foods on the microbiome and specific key gut and other health biomarkers in local populations. Really, the key focus is the application of these fermented foods as a novel maternal nutrition intervention, and really providing local investigators in sub-Saharan Africa and South Asia specific tools and resources to build the capacity to study and really leverage these traditional processes as next generation novel health and nutrition interventions. And so really the challenge breaks up across three specific areas. The first is identifying a specific local fermented food for study. The thought here is that many of these fermented foods, even some that may not be considered fermented foods, have living microbes that transform the underlying substrate. And so the first step is really identifying a specific local food that has potential, is consumed by many people, but has potential as a maternal nutrition intervention. One key criteria here is that the focus is really on fermented plant-based substrates or fermented vegetables, given that given the low cost of those substrates themselves. The second part of the challenge is really focused on then applying and using that food in a small pilot scale longitudinal intervention study. And so the study we just saw from Erica just a few presentations back, is really a exciting template for these sorts of studies where individual kind of fermented food or food-based substrate can be studied in really high density in a really controlled trial. And here the goal is basically understanding the specific effect of the fermented food versus a naive population, basically no consumption of this fermented food in a small cohort population of women of reproductive age. And the final part of this challenge is really then understanding how these fermented foods, these traditionally consumed foods, can ultimately impact the underlying structure and function of human health and the human microbiome. And so this involves biological sample characterization, understanding how the gut microbiome changes before and after the fermented food intervention, but also looking at key biomarkers, things like inflammatory analogs for inflammation in fecal material, as well as blood sample characterization before and after the intervention. Next slide, please. And really, we think that sequencing is really a democratizing and enabling scientific tool in the pursuit of studying these fermented foods, as well as how they impact the gut and gut health. Really, over the last 5 to 10 years, sequencing has enabled us to, for the first time really in human history, measure all of these microbes, both within humans as well as in the environment. And so a key part of this, this grand challenge is also providing access to specific sequencing infrastructure and methodology at these local geographies and for these local investigators. And that is a quick overview of the challenge, and hopefully we have a little bit of time to dive into some questions around it. Thank you.
Ravi, thanks so much. Really helpful to lay out the specifics of the challenge. I think that will come in really handy for people that are applying. As a next presentation, it's my great pleasure to introduce Ashish Jha. He's an assistant professor of biology at NYU in Abu Dhabi. And there he leads the genetic heritage group and studies human microbiome interactions. And he will describe how the call aims to help develop in-country sequencing capacity, and through a research network approach, align and synergize rigorous, well-designed pilot trials of diverse fermented foods across different geographies. Ashish?
Thank you, Chris. I am keeping my video off so that because of the internet bandwidth problem here. But thank you for the introduction, and it's been a privilege to be a part of this effort. So as Ravi mentioned, fermentation is a human cultural heritage, and humans learned to ferment food thousands of years ago, and there is tremendous diversity of, we can go to the next slide. There's a tremendous diversity of fermented foods in many cultures across the world. Here's a snapshot of different types of fermented foods from one country, Nepal, the country that I'm from. And there are many, many more types of fermented foods in Africa as well as in Asia. These foods are truly superfoods, but we really need to understand how they influence our health in a positive way. So here we can see fermented grains, fermented vegetables, as well as cultured dairy. Next slide. So studying the microbes of fermented food presents excellent opportunities, but it also comes with several challenges. First, how do we sequence the microbes of different types of fermented foods so that we produce consistent and reliable results? Second is, how do we compare these results across studies to get a better understanding of microbes and their function? And third is understanding how these microbes influence the different aspects of human health. So to answer these questions, we really think that the standardizing, we need to standardize how we do microbiomics of fermented foods. And there are several aspects of fermented foods that we think need harmonization across studies. First, we need to understand fermented foods and how they're used. So to address that, we are currently developing a survey questionnaire that can be used across geography to understand different types of ingredients that are being used in fermented foods. So it could be grains or vegetables or dairy or whatnot. The next thing is the different types of fermented methods. As Ravi was mentioning earlier, there are many different types of fermenting methods, adding salt, adding vinegar, or just like adding water. And the duration of fermentation may vary as well. Some of them are short-term, days to a few hours to a day, a few days. Some of them are longer term, going from weeks to months. The way we consume fermented foods are also different. Sometimes we consume them raw, and sometimes we have to cook them. And when we consume the fermented foods could also be different. For example, the beliefs and practices around eating fermented foods can vary between different population. And also when we can eat those food, who can eat them also can differ. For example, can pregnant women eat some fermented food or not? Whether children can eat certain food or not can vary. Second is sampling and processing. Once we figure out which fermented foods we want to study, how do we sample them? How do we process them so that the data can be compared across food types, across labs, and across geographic region? It is a challenge. So to address this, we're establishing best practices in sample collection, sample processing, storage, handling, and every aspect of sampling in the lab to obtain quality sequencing and other types of omics data. Third thing is sequencing itself. So amplicon sequencing is probably the easiest and economical method of sequencing. Again, for the comparative purposes, it is important to harmonize what target region we're trying to amplify, what type of sequencing methods we're using, what type of platforms we're using across studies. Because only if we use a consistent approach, we can actually start to compare data across studies and make sense of how the microbes are functioning. And then there's also data analysis. We are excited to generate data, but generating the sequence data is only half the problem. And after the genomics data is generated, we need to analyze it, and that can be overwhelming. So to address that, we're currently developing analytical workflows for microbial sequencing data. And all the things that I've described here is only the first step, and there are several subsequent larger scale genomic analysis that will need to be conducted to completely understand the human microbiome interaction. So there's plenty of room for future collaboration in metagenomics, metabolomics, metaproteomics, and downstream. Next slide. So what we're doing right now is I'm working with the Sonnenberg lab at Stanford to develop a complete pipeline of methodologies spanning from sample collection to sequencing and data analysis workflows. The methods developed by Stanford and optimized at NYU AD will be translated to standard operating protocols that will be shared with the grand challenges awardees. This will allow us to integrate data across different experiments as well as labs. We can also, we will be able to integrate previously published data from different studies. We can, NYU AD will provide virtual trainings to assist awardees in some design aspects of the project, sampling, processing, sequencing, as well as data analysis in their own countries, wherever the awardees are coming from. The primary goal of this effort is not only to facilitate in-country genomics data generation, but also to provide some sort of a support system for local researchers to share their concerns, problems, and also celebrate their results. The awardees can then use the preliminary amplicon sequencing work and develop future projects within themselves, as well as with Stanford and NYU AD or academic or other academic and industrial organization of their choice. So next slide. So in summary, we recognize that studying fermented food is very important, and we want to harmonize different aspects of studying fermented foods. And Stanford and NYU AD will develop protocols that the awardees can use. We'll conduct virtual trainings to assist awardees in different aspects of this work. And we hope to develop future collaborative projects with the awardees. With that, we can go to the next slide. I just thank you for your attention.
Thanks, Ashish, and thanks all for your very rich talks. That concludes the presentation portion, and would like now to transition to the Q&A. So we'll hand the baton over to Shelby.
Thanks, Chris. So I'm going to go down the list of the questions that have been submitted so far. And if I ask your question and you want to expand on it or add anything, please feel free to chime in. And then I'll just ask whomever is most appropriate to answer the question would also chime in with a response. So first off, for Chris's presentation, this question I believe was for Chris's presentation. Is it important to have fermented food to be rich in protein? How about fermented rice water or bran water?
Interesting question. So at the end of the day, microbes use all three primary macronutrients. So that's protein, it's carbohydrates, and it's fats in their fermentation processes. Although perhaps disproportionately, it's the carbohydrates that are contributing as substrates for the microbes. In terms of protein, there's it's a good question, and the degree to which protein, high protein, is relevant in fermentation, and maybe others on the call have some thoughts there. In terms of rice bran, also a very good question, and independent of fermentation, there is work in the pediatric context in LMIC showing that rice bran is impactful in growth. And so the possibility of combining rice bran with fermentation is an intriguing question. Did others on the presentation panel have specific thoughts around microbes and protein?
I can make a quick comment. I do think there's a lot of exciting opportunities around improving the quality of protein. So many staple crops are deficient, like let's say something like corn or rice, are deficient in things like lysine or other key amino acids that are really important for growth and other key parts of maternal and infant nutrition. And so both in these existing kind of staple crops, but also some of the emerging really commodity protein sources, things like pea or legume protein, the quality of the protein and amino acids themselves is very much a concern. And so it has been shown that fermentation can improve like the concentration of some of these key amino acids. And so I think it is potentially a really interesting avenue by which fermentation can improve the quality of some of these really staple protein sources in the food chain.
Great, thank you. And if there's nothing else to add to that question, I'll move on to the next one, which is directed towards Erica. Was there any benefit from eating just one type of fermented food, or did participants, did participants need to eat a variety of fermented foods?
Yeah, that's a great question. I mean, we saw participants do lots of different things. Some participants really focused on one type of fermented food and ate a bunch of it, and others ate a variety. Keep in mind, our N was quite small, only 18 participants, but it didn't appear that seemed to make a difference. We did see this cohort-wide effect of consuming fermented foods. The nice thing about the way the study was set up and how we're thinking about future studies is sort of a reverse translation approach. So now that we understand a little bit of how fermented foods works in humans, we can build model systems in mice where we can ask these questions specifically. just providing one type of fermented food, a bunch of it, versus providing a variety of fermented foods in mouse models and see if that really has an impact. But at least in the small study that we performed in humans, it didn't really seem to matter.
Great, thank you so much. The next question is for Ravi. So with so many different substrates or foods and so many different paths fermentation can take, i.e. different consortia, strains, different temperatures, etc., what is the path forward for studying these products reproducibly and determining what is optimal for human health?
Yeah, I think a great question from Justin. I think there's two parts to this. So like the first is basically defining really key biomarkers or analogs, because these foods are impacting a really complex underlying system, human health, digestion, nutrition. You really have to define really specific key biomarkers that are endpoints for these sorts of studies. And so for this Grand Challenges call, we identified a few. It's somewhat open-ended, going across nutrition, things like iron and B vitamins, into inflammation, which has been shown to kind of associate with maternal nutrition and birth outcomes. So I think the first part to this, things like lipocalin-2, IL-6, CRP, in fecal and blood samples. But so I think the first answer to this is really being clear and trying to define very specific end biomarkers that can be the outputs of some of these very complex studies at the end of the day. I think the second regarding kind of the complexity is really thinking about highly densely sampled longitudinal studies where individual people serve as their own controls. And so I think having studies where you can measure what is like the biological state of a microbiome and the biological state of health before an intervention and after an intervention becomes really powerful in being able to specifically see how people change over time, because many of these effects may be personalized or may be dependent on many other variables. And so that really has to be controlled for in the design of these studies.
Great, thank you so much. So I'm going to go on to a question that says, thank you so much for this amazing set of talks. Regarding the fermented foods proposal, what could be the scope for creating a biorepository of fermented foods and constituent strains / bioactive molecules to move beyond sequencing to further functional characterization of fermented foods, to be safeguarded for future generations, and to enable testing mechanisms, generalizability of health promoting effects?
So I'm happy to take that one. The real focus of this call or grand challenge is to look at the impact of a chosen fermented foods on biomarkers of inflammation and microbiome in women of reproductive age setting. And that should be the thrust of the proposal. In terms of characterizing the broad scope of fermented foods that exist within particular geographies, while that's not the focus of this grand challenge, recognize that that is a valuable step forward within fermented foods as well. But really, the idea is to take a possible sort of perceived best shot on goal of fermented foods based on what we do know within a particular geography and evaluate that in a small pilot context.
Great, thank you. And we just have a couple more here. So quantifying and classifying fermented food intake in non-observed settings like the home seems like a challenge. Would the foundation be interested in approaches to better capture what, when, and how much study participants are consuming at home? How much study participants are consuming at home?
Yeah, and happy to take that as well. I think the answer is probably similar to the previous question. And I guess in so much as the particular food that you're evaluating within a small pilot, if there was interest in doing a little bit of descriptive work on baseline consumption of that food, I think that would be valuable.
Great. And this one is directed towards Erica. Did you look for the specific micronutrient, such as provitamin A or vitamin B, producing bacterial strains as well?
Yeah, so we didn't look at that specifically. We were mostly interested in just an overall effect of consuming fermented foods. We have done some other studies looking at specific probiotic strains. And so you could imagine doing studies like that and testing specific strains and how they behave. One thing that I will say is that when we did our probiotic strain study, we didn't see the profound effect that we did see in fermented foods. And so we do think that there may be an aspect of fermented foods, be it the maybe the metabolic products that these microbes are producing within the fermented food, that might be an important biological effect of the fermented foods that we did see.
Okay. And I have one more question, Chris. If you feel like there's time, I can go back to, if we're okay going a little bit longer, I'll go ahead and ask it. Given human coevolution with fermented foods, do you think different human populations will have specific local fermented foods to which they are more adapted? i.e., are local fermented foods likely to have a larger and more positive health impact than an imported fermented food? You're on mute, Chris.
Oh, yeah, I thought maybe let somebody on the panel ponder that one.
I'm happy to answer that, even though it was asked by my husband. So maybe we would see it in a very similar way. You know, from studies of the microbiomes, looking at, so there was a study that came out in Asian populations and found that these people's microbiome harbored a strain of microbe that was able to digest porphyrin, which is a complex carbohydrate found in seaweed. And so there was an indication there that the microbiome had really adapted to the specific carbohydrate landscape of these people's diet. And so I think that it may be something that we would expect with fermented foods as well. If there is a long history of consuming a certain type of fermented food within the population, I think that it is entirely possible that the microbiome has adapted to expect that fermented food consumption. And it's possible that consuming an outside fermented food that isn't a part of the history of the diet of these populations, we may not see as strong of an effect as a locally fermented food, but it really is an open question that would be an interesting one to address.
Thank you so much. And Chris, as I turn things back over to you to close out, I did want to just flag, there is a question that came up from a number of people just wondering if the slides could be shared afterwards. And so I wanted to pose that to you as I turn it back over.
Absolutely. I think it would be wonderful if we could have a link from the Grand Challenge site, possibly to the slides. So anyone that's interested in the call will have access to this really rich discussion. So that's a resounding yes. Recognizing that we're about 10 minutes over, I thought valuable to capture some of these really good questions. I did want to thank everyone for joining and really look forward to seeing your submitted Grand Challenge proposals. Thank you.